Welcome to Methodical Medics. This is our first video, and we're starting with one of the most important pathways in biology, glycolysis. The word glycolysis literally means to split sugar. And in this video, we'll break down how glucose is converted step by step into pyruvate. Understanding this pathway is crucial because it's the first stage of cellular respiration and the foundation of how cells harness energy in the form of ATP. Each step of glycolysis occurs in the cytoplasm, which is represented by the blue background. It begins with glucose, a 6-carbon sugar which we get from digesting foods. Adenosine triphosphate is also needed to begin glycolysis, which is why this is known as the investment phase. The red rectangle represents an enzyme which catalyzes the reaction. In this case, it's hexokinase. What hexokinase does is it transfers a phosphate group from ATP to the 6th carbon of glucose. And as a result, ATP is converted into ADP, and our product is glucose 6-phosphate, the first intermediate of glycolysis. Also, take note of the reaction equation. The arrow is unidirectional, which means that the reaction is thermodynamically favorable towards glucose 6-phosphate and is irreversible. There are 3 irreversible steps in glycolysis, which are often points of regulation. In the second step of glycolysis, glucose 6-phosphate becomes isomerized. This is catalyzed by the enzyme phosphoglucose isomerase, and the only change is in arrangement, converting the ring from hexagonal glucose into pentagonal fructose form. It still has 6 carbon atoms, and the new intermediate is fructose 6-phosphate. This is important because it sets up the molecule for the next phosphorylation step. This time, the reaction is in equilibrium, which means that it is reversible and can convert to the fructose form back into the glucose form, depending on what the body needs. Step 3 is another investment step, requiring another molecule of ATP, and is very similar to step 1. This reaction is catalyzed by phosphofructokinase 1, often abbreviated to PFK1, and has a similar role to hexokinase. It takes a phosphate group from ATP and transfers it to the first carbon of fructose 6-phosphate. This forms ADP again and fructose 1,6-bisphosphate. This step is irreversible and is considered the key regulatory step of glycolysis, because once fructose 1,6-bisphosphate is formed, the cell is fully committed to completing the pathway. Steps 4 and 5 will be shown in one animation because they both lead to the same intermediate. In step 4, the enzyme aldolase cleaves fructose 1,6-bisphosphate into 2 3-carbon molecules. The first molecule is glyceraldehyde 3-phosphate, or G3P. The second molecule is dihydroxyacetone phosphate, or DHAP. Notice the oxygen positions. In G3P, the oxygen is attached to the first carbon, while in DHAP, it's attached to the second. Only G3P can directly continue into glycolysis. So to prevent DHAP from building up, the enzyme triose phosphate isomerase converts DHAP into another G3P. By the end of this step, we have 2 molecules of G3P ready to continue. This also marks the end of the energy investment phase of glycolysis. In step 6, glycolysis officially enters the payoff phase. Remember that steps 4 and 5 gave us 2 molecules of G3P, so everything from here happens twice. In this step, glyceraldehyde 3-phosphate reacts with NAD+ and an inorganic phosphate group. NAD+ is the cell's electron carriers and is in an uncharged state. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes this reaction. NAD+ is reduced to NADH, capturing high-energy electrons. And at the same time, it transfers the phosphate group into G3P, forming 1,3-bisphosphoglycerate. This is a redox reaction. G3P is oxidized while NAD+ is reduced. In total, 2 NADH molecules are produced here, which will later be used to help generate ATP. Step 7 is where glycolysis produces its first ATP and is catalyzed by the enzyme phosphoglycerate kinase. 1,3-bisphosphoglycerate is a high-energy molecule, and it donates one of its phosphate groups to ADP, forming ATP, and 3-phosphoglycerate. Remember that this step produces 2 ATP. Step 8 is relatively simple. Phosphoglycerate mutase shifts the phosphate group from the third carbon in 3-phosphoglycerate to the second. Therefore, it becomes 2-phosphoglycerate. It's simply a structural isomerization. In step 9, 2-phosphoglycerate is converted by the enzyme enolase. During this reaction, a water molecule is removed and a double bond is formed. This type of reaction is called dehydration, and as a result, a high-energy compound is produced called phosphoenol pyruvate, or PEP. It is now primed to transfer its phosphate group in the final step of glycolysis. In the 10th and final step of glycolysis, phosphoenol pyruvate and ADP enter pyruvate kinase. This enzyme transfers the high-energy phosphate group of PEP to ADP, which produces ATP and the last product called pyruvate. This step is the third and final irreversible step in glycolysis. Remember that the step produces 2 ATP and 2 pyruvate molecules. Pyruvate is the end product of glycolysis and will move into the mitochondria for the next stage of cellular respiration. Now let's summarize glycolysis in a simple way. We start with one glucose molecule, a 6-carbon sugar, and 2 ATP molecules to get the process going. Over the course of 10 enzyme-catalyzed steps, glucose is broken down into 2 molecules of pyruvate, each containing 3 carbons. Along the way, glycolysis generates a net gain of 2 ATP molecules and 2 NADH molecules, which store high-energy electrons. These products are critical because the ATP provides immediate energy, while NADH and pyruvate feed into the later stages of cellular respiration. Thank you, I hope you enjoyed, and I can't wait to see you in the next video about the bridge reaction.